Let me start by saying hi to everyone, and also to thank the organizers for this meeting, Daniela, Alex, Martin. I think this meeting has always been great, but now it's really becoming truly spectacular, so thank you. I want to start on a bit of a sad note by acknowledging a friend and a colleague, Judy Campisi, that many of you know, who sadly left us early this year. And not many of us actually very few of us actually have the privilege of not only, generating new interesting data, but also creating a whole field. And she was really instrumental in the concept of senescence. She discovered the first biomarker for senescent cells. She discovered the SASP. She invented the concept of senolytics, and so we miss her very much, and I know that she would be amazed to see what the field has actually become, and will continue to become. So. Okay, on to the science. Half of my lab is actually focused on trying to understand the biology of immune aging. And we decided to focus on the immune system for a very good reason, is that our hypothesis is that aging in the immune system could be a universal driver of organismal aging. If you think about organs that are globally distributed in any mammals, for example, we have the nervous system, the endocrine system, and the immune system. So the immune system is actually distributed across the whole organism. So whatever happens in the immune system has the potential of affecting pretty much every cell in the organism. And this was beautifully illustrated in these two papers, which actually got us really excited. What both of these papers did is to generate an immune-specific lesion, either by deleting the protein called TFAM, which is the major mitochondrial transcription factor, or another protein, ERCC1, which is involved in DNA repair. And using specific Cre drivers, they were able to delete these proteins in immune cells only. And the remarkable finding of both of these papers is that these specific deletions in the immune system lead to organismal aging. And this was best illustrated, I want to make sure that this best illustrated by the fact that these animals live shorter in both cases, so the global life expectancy was decreased, but they were also able to demonstrate an increase in senescent cells in non-immune cells, so what actually Judy and others have called secondary senescence. So really this is one reason why we are focusing on the immune system. First, it is the most accessible tissue in humans. We can actually do pretty much anything we want in terms of deletion, transfection, and so on. But also by sampling the immune system in humans, we can actually assess directly, without any sort of dramatic interventions, the state of immune aging. And so all of our focus actually has been on trying to understand the following questions. What happens to specific immune populations during aging in humans? Is there such a thing as immune cell senescence in human? There's a confounding variable here, is that a lot of people are talking about immunosenescence. They are referring to aging in the immune system. They are not referring to senescence. And so that has created a lot of confusion. And so I never use, by the way, the term immunosenescence, and I think we probably shouldn't, because it generates a confusion in terms of what we're studying. And I talked about this last year and actually was able to show you, hopefully, and to convince you that there is indeed some low-level senescence in the immune system of humans as they age. And second, it is not measured by beta-gal. And I think I'll say this again one more time, because there are a number of clinical trials that are ongoing right now for senolytics, where the readout is using SA beta-gal, FDG, spider-gal, and all those. Those are not measuring senescence. They are measuring lysosomal dysfunction. So that's all I will say about this today. The key thing, another two key questions that we are trying to address is how do we measure human immune aging? And I'll be focusing a lot of my presentation on this. And last, I think the true potential of this field and what we're doing is we believe this is one of the rare experimental system in which we can test interventions in an in vitro system using primary immune cells. And you've all heard about the X Prize. And so one of the approaches is to use these in vitro systems based on primary human cells to test a variety of interventions and see whether we can actually modulate the rate of aging in vitro. So a word about the immune system. Once you draw blood from someone's arms, there's an incredible complexity. Two big arms of the immune system, myeloid cells and lymphoid cells. Today, using the technology we have in the lab, we can identify 640 different populations in the blood. So the complexity is staggering, but it is also a reflection of the fact that we have to take into account these variation in populations. Our major focus has been on lymphocytes for a number of reasons. First, we cannot study everything, and we think that the lymphocytes by themselves represent a unique frailty point in the immune system in terms of aging, because most of your T cells are generated in the thymus, and we know that the thymus involutes in most people around age 50. So past age 50 or 55, depending how well you are aging, most people will rely on a pool of naive T cells that was generated early in life. And therefore, we think represents really a key choking point in terms of aging of the immune system. Let me show you also one additional slide of introduction about what are T cells. There are two broad categories of T cells, CD4 and CD8. They are, as I mentioned, naive T cells are the initial precursor. They're generated in the thymus. And they differentiate through each of these cells carries a single T cell receptor, which recognizes a single antigen. And this is where the diversity of the immune system comes from. As these cells meet their cognate antigens, they will become activated and undergo a set of proliferation. So this is the basis for a vaccination response. Once you vaccinate someone, you pick out a few clones that are recognizing a unique antigen, you allow them to proliferate and to persist. And so these cells persist in the form of memory T cells, central memory T cells. If they meet their antigen again, so you've been vaccinated, you have central memory T cells, you meet the antigen because you get COVID, these cells become rapidly amplified, and this is the basics of an effective immune response. Now, another reason why it's important to study the immune system has been really sort of dramatically illustrated recently with the COVID pandemic, where we know your risk of dying is close to 200-fold higher if you're above 80. And a lot of noise has been made about the risk factor, age being a risk factor for death by COVID, but this is not unique to COVID. The same thing happens in terms of influenza, RSV, and I suspect many other viruses. So get your vaccinations. Now, the other thing that we should remember also is that by the time you reach 70, only 30% of people are able to mount a successful immune response. And so, someone has asked me, well, why should we have everybody being vaccinated? This is because we're vaccinating the whole population, so we're limiting the spread of the virus, and we're protecting the people who are above 70, who are most likely unable to mount this immune response. There's also an incredible potential in reestablishing the ability of older individuals to respond to a vaccine or to an infection. So a lot of the initial work, so this is a reasonably new project in the lab that I started when I joined the Buck Institute. One of the first things we did was to purchase the latest technology in terms of flow cytometry. It's called the spectral flow. It's shown here. This is technology, as I mentioned, that allows us to detect close to more than 600 different populations in peripheral blood. So the first thing we did is to actually revisit some really simple and old experiments of what happens to all of these populations during aging. And on the left, I'm showing you what we call a dimensionality reduction plot, which is essentially taking all of this data and collapsing it into 2 dimensions, which shows you that there are a number of different populations corresponding to B cells and K cells and so on. And then we can look in a population of people aged from 20 to 90, what happens to each of these cells. And we were able to sort of repeat what has been known for a long time. Your NK cells progressively increase, so do your monocytes. Your CD8 T cells decrease, and so do your gamma delta T cells. More interestingly is as I mentioned, we couldn't focus on everything, so we have intensely focused on CD8 T cells and CD4 T cells. And so this is now another dimensionality reduction plot focusing only on the CD8 T cells. And you can see we go way beyond what had been known and appreciated before in terms of the resolution of all of these different subpopulations, so that we can then look at them and say what happens to each of them during aging. And again, the dramatic decrease in naive T cell is really something that gives me pause when I hear anyone talking about immortality or any outlandish claims about what this field is going to accomplish. I said, first explain to me how we're going to solve this. There's no more thymus. Maybe we'll generate thymus regeneration, but this is obviously a huge aspect of aging. By the time you reach 80, most people do not have any naive T cells. Remarkably, we see the converse. There's other population, the memory T cells, which are a reflection of what happens in terms of which T cells carrying which antigens have been activated. You can see an increase in these populations during aging. What was actually really unexpected, and hopefully this will be a full talk next year because this is something we're actively working on, we found a population, a novel population which had not been recognized before, called CXCR3 minus central memory T cell, which is essentially absent in young people and dramatically increases during aging, but not in everyone. You can see some people actually have very low levels. So we are, without getting into the story, we are in the process of characterizing them for the immune system aficionados here. These look like Th2 cells. So these are CD8 T cells that express a variety of interleukins, such as IL-4, IL-5, IL-13, and they seem to be, preliminary data seems to be showing that they are associated with some specific disease. Very early preliminary data, so I don't feel comfortable showing them, but we believe that they could be an important aspect of some of the chronic disease of aging. So the rest of my talk will be about epigenetic clock. I realize I'm running out of time. I don't need to introduce them in terms of what they do and why we all use them. Again, one of the findings that I showed you last year is the fact that if we purify these individual populations of T cells and we measure their epigenetic age using the existing clocks, and here we've used Steve Horvath clock or PhenoAge and so on, in almost all cases we find the naive T cells are 15 to 20 years younger than the individual. And in contrast to the effector memory T cells and the TEMRA, the terminally differentiated T cells, which are actually look typically older than the individual. I think what's really interesting about this is that the immune system being sort of distributed and not a solid tissue, we really are able to do things that most people in the field are not able to do. And I would predict that in most tissues, your stem cells are likely to look much younger than your terminally differentiated T cells. With the immune system, we can look at this. Now, what the implication of this is that, as I've just showed you, the relative proportion of these cells changes during age. You lose your naive T cells, which look younger, and you get more of these cells which look older. So this could be a simple reason why we see actually an effect of age on your epigenetic markers. And this is illustrated here in the best way. So what the data showed is that these epigenetic clocks that are existing today actually are measuring two things. One, they're measuring a change in population proportions, but they're also measuring what we call intrinsic aging, which is each of these cells. If you look at the naive T cells as a function of age, you can see age here and the predicted age. You can see that there is a signature of aging in naive T cells, but the signature is different in the memory T cells. They are globally older. So what we've been after is trying to identify what we call intrinsic aging, which is not a proportion of T cells, but what the naive T cells, are they changing during the aging process? And so with Alan Tomusiak, a graduate student in the lab, we were able to generate a new clock that we call Intrint clock, which essentially we removed all of the CPGs that were associated with differentiation. And so, and I'm going to show you briefly the data. The paper is accepted, and it's in press if you want to take a look at it. The take-home message here is that this intrinsic clock actually does not change anymore during T-cell differentiation. You can see this across a whole variety, not only in T cells, but also in B cells and others, which actually is quite interesting. So we've been very busy trying to understand what does it actually measure. I can show you here data showing that it is sensitive to reprogramming. When we reprogram other cells, you can find that the clock measures a complete reversion to H0, and it's also sensitive to a senescence in vitro model. So potentially functioning as a marker of senescence. Now, we use then this new clock to go and revisit some of the findings that frankly had disturbed me, and from a purely sort of intellectual standpoint. There were a number of papers claiming that COVID-19 infection was associated with a 15-year acceleration of aging. I cannot understand how this would happen in 3 weeks, but a number of papers reported this. Now, what happens during COVID infection is you have a massive lymphocyte activation and amplification, so you're shifting acutely the ratio between your naive T cells and your memory T cells. So we went back to this and actually were able to show using the new clock that we saw no effect of HIV infection. There also were papers claiming that HIV infection was associated with an aging acceleration, so we don't see an effect, and we don't see an effect in terms of the intrinsic clock, in terms of COVID. Now the question is, what is this clock measuring? And I just want to share with you some early data which shows a number of diseases that are linked with intrinsic clock acceleration, dementia, Parkinson's, Huntington, many forms of cancer. But remarkably, many of the diseases that we have been told are associated with accelerated aging, such as HIV, rheumatoid arthritis, multiple sclerosis, celiac disease, and so on, many of these diseases are characterized by chronic immune activation. And so I think it's important for the field. I'm not claiming that these diseases are not deleterious or causing any problems, but I think it's important that we try to understand exactly what we are measuring. So the next thing that I want to, last thing I want to discuss in the next couple of minutes is preliminary data that we're in the process of preparing for submission, trying to generate another immune clock. And to this, one of the problems that we've had, and I think everyone has in the field, is when you generate a clock and you have a number of CPG sites that you are using, the question has always been, why are these CPG sites changing? What are the genes that are associated? What does it tell us about the biology of aging? And the answer typically is, you cannot really figure out, you cannot see a signature of many of the pathways that we know are regulating aging. So that's a problem. One of the reasons for this could be the fact that, for example, the early Horvath clock is actually a mixture of two different clocks, a cell composition clock and an intrin clock. That could be one of the problems. But even the intrin clock gave us limited amount of information in terms of the genes that are important. So we decided to go through another step, which is to generate a single-cell transcriptomics. The idea here is that single-cell transcriptomics brings you one step closer to actually what the proteins are doing, obviously looking at transcripts. So the idea was to try to gain, not only to have an additional marker, but also to gain unique biological insights from differential gene expression in aging. So the way we went about this, again, because we're dealing with on peripheral blood with an extremely complex and heterogeneous population, was to generate a cell composition, a cell prediction algorithm using a single cell transcript. And so this, and I'll mention this right at the end, one additional idea of why we should generate an immune single cell transcriptomics clock is the idea that I mentioned right at the beginning of my talk, is that this could be used in an in vitro screening system for interventions in the future. So the data set that we use actually was already in the literature, and I want to acknowledge the work of Maxim Artyomov here, who generated a single-cell atlas of healthy human blood using a large number of more than 300 samples. So the data was available online, so we were able to go and study this. Parenthetically, this group, using single-cell transcriptomics, were able to identify the same population, these type 2 memory T cells that I mentioned are, that we identified using flow cytometry. So we have now 2 different congruent pieces of data that suggest that these cells are age-associated and obviously warrant further characterization. So I won't have the time to go through all of the data. The paper is in the last stages of being prepared. You'll see it on bioRxiv pretty soon. What we did is first, and this is work done by Alan Tomusiak and Sierra Laura, who is a student in the lab, who's actually attending the meeting, and a student with Morten at Copenhagen University. So thank you, Morten. So we first did a pre-filter on all age-correlated genes. So out of all the transcripts, we selected those that actually showed some type of correlation with aging. We trained the model to predict one of 6 different cell types, and then generated this clock. And again, I don't have a lot of time. I just want to show you that the cell type prediction actually really maps with what we would expect. So it's highly precise and correlates with other ways that we can look at these different T-cell subsets. And here's a single piece of data that shows you that the clock, indeed, this is the clock on the training set and the clock on the test set. And you can see it's not the most precise clock, but it might be, again, a reflection of the fact that there's a lot more heterogeneity in the population in terms of immune aging. I'm almost done. The last thing I want to do is to show you sort of a Go analysis of some of the transcripts that we found to be strongest predictors of aging in the immune system. And this is actually the ribosome. And so won't be able to go through any kind of the details of the data, but it is highly reminiscent of what some other people have found in other experimental systems. So this is an interesting finding. Another finding that we found is a really strong correlation with an elongation deficit at the transcriptional level, which has been shown by others. We find this in aging T cells as well. So I want to finish by repeating very briefly, a unique subset of CD8 T cells specifically accumulates during aging. Most epigenetic clocks measure both immune activation, not inflammation, this is distinct, and intrinsic immune aging. Our new clock, Intrin clock, measures immune aging independently of immune activation. By the way, we've shared the algorithm with a True Diagnostic. I haven't checked with Ryan if he's here, whether he's willing, to. I don't know if he's providing it as part of your analysis with True Diagnostic, but we want the clock to be used. We think it brings a whole other dimension. And finally, a new single-cell transcriptomics clocks highlights molecular aspects of aging. It can be used to measure aging, but also to test aging interventions in primary human cells in vitro. And last but not least, I think I've acknowledged many of the people in my group that functions on work on immune aging, in particular Alan Tomusiak, who's here as well, and Sierra Laura. Thank you.
Well, thank you, Eric, for the great talk. We have time for one quick question from the audience before we go to the next speaker. Right at the back, right there. Or we can go first. Please make it quick.
Yeah. Excellent talk, Eric. The intrinsic clock is amazing. My question is about your analysis around HIV infections and COVID specifically. So when you say it's measuring immune aging, yet it's not really capturing changes in HIV and COVID, we know that HIV individuals, HIV-positive individuals specifically do have increased incidences of cardiovascular disease. They do have increased sort of systemic inflammation, but you're saying that they do not have increased biological aging. So I'm just trying to reconvene, think about both of those ideas together. What are your thoughts about that?
The way to think about it is, and this applies not only to HIV, but also applies to rheumatoid arthritis, which has been claimed as a disease of accelerated aging. And I think, if the data says that this is not truly aging. There might be an increased risk of a whole series of disease, and this might be linked to chronic inflammation and so on, but at least most of what we're saying is not that these people do not show some of the manifestations of aging, but when you look at their immune system aging, this is not what it looks like. It looks like a chronic immune activation. Now, immune activation by itself might become a source of aging. That I totally agree.
So it's a quick question, but it might be a long answer, but I'll put it out there anyway, which is the immune, great talk, by the way. The immune system has a bit of a bad reputation in our field, like with inflammation. So what about experiments just getting rid of it, right? I mean, there are, for example, immunodeficient mice. As far as I'm aware, they have pretty normal lifespans, but maybe you could elaborate on that.
Well, if you have no immune system, you have to live in a bubble, and that's the answer, So, I mean, that's the short answer, in effect, that the kids who have the equivalent mutation, the SCID mutation, these mice that are completely immunodeficient, these children have to live in a bubble, and they cannot survive, 2 weeks outside of the bubble. So, the key, another way to answer your question is maybe more sort of subtle, is the idea that we know that chronic immune activation is a driver of aging, okay? There's clear indication, and we've been able to link many of the manifestations of chronic immune activation with NAD depletion, for example. That's I think one of the big ones. So what hasn't been answered is, what is the driver of this chronic immune activation during aging? Is it the aging? I'm convinced it is actually the aging process, because the immune system recognizes any damage, be it induced by a virus, by a bacteria, or tissue damage. And so there's a big question of so if you were to completely eliminate the immune system in someone old, you might eliminate the bad secondary effect of immune activation, but you might also not be removing senescent cells, for example. That's part of the immune system. We know that they are constantly being removed by the immune system. So it's a really intricate question, and the short answer is that no, immune suppression is not going to make us live longer. I think it would actually probably shorten our lives.
Thanks, Eric.